Very big hello to my wonderful PU2 champions. I hope all my dear children are doing absolutely great. So few of you requested me to cover the biotechnology related chapters first, which is why I'm here. I finished off one chapter, that is biotechnology principles and processes, and now we are here with the next chapter, that is biotechnology and its application. But most of you find it to be difficult, but let me tell you, it feels difficult for you because probably it's because you have not come across this topic during your entire schooling. But in general, if you try a bit to study this chapter, it's going to be super easy. And please do not worry, I am here to help you out. And if at all you are someone who has a doubt that, ma'am, I have not studied this chapter actually, will this video be useful for me then, because you are directly solving questions, right? But the answer to the question is that yes, you can still rely on the video, because my dear children, not just me but any teacher out there thinks that as of now the person in front of me might not know everything about it, and I have to explain in a way that they understand it from the basics. So obviously when I show you a question, I'll make sure that you understand the question properly, each and every terms of it, and also understand the concepts. So this video is going to be super helpful for you, super important to you as well, because I've taken questions from the recent model question paper, from the revised question bank, and also I have included the previous year question paper, and I can guarantee you that you will be getting those 6 marks that you can get from this chapter according to the blueprint 2025-26. So this chapter gives you 2 questions, one from the 1 marker category and one from the 5 marker category, and if at all you are able to crack it, it's going to be super helpful for you to grab those 5 marks easily. Yes? And I'm pretty sure you're gonna do that because you guys are rock stars. So let's go ahead with the model question paper question. This is for the year 2025-26. I've shown you the question and here is the question number. So this is from model question paper 1. Question number 12 we are covering. As I am always telling you, whenever you watch my video, make sure you having that model question paper. If not the soft copy, if it's not the hard copy, at least the soft copy, and put just a tick mark out there with the help of a pen, okay? In your computer. So you can do that to understand that, okay, I am covering all the model question papers also simultaneously, and you will be very much relaxed then. So are we ready to begin with the first question? Let's go ahead. So given below are the steps in the gene therapy of a patient with adenosine deaminase deficiency. What? These are the steps in gene therapy of a patient with what? Adenosine deaminase deficiency. First of all, what is that? So adenosine deaminase is an enzyme. It is an enzyme, and this enzyme sole function is to basically break down something that is toxic for the cell. So it is detoxifying the cell. Okay, its sole role, it is to detoxify the cell. Now, who needs this particular enzyme the most? There is one group of cells in our body that is known as WBCs, the white blood cells, which is there for immune system, which is there for increasing our immunity. These white blood cells, there are a type of it which is known as lymphocytes. They are known as lymphocytes, and they are the actual one who do all the fighting scenes. Now, the lymphocytes are the one that needs this enzyme the most, because there are probability in them, if at all the toxic substance stays, it can cause bigger problem. Now, suppose there is a condition where in this particular cell, in our normal cells, the gene that codes for this enzyme is not there. It is missing. There is a deletion happening. There is a deletion happening, which is why this enzyme gene is not there. That means my enzyme is not being produced. That means the toxic substance is getting accumulated, which initially means that my immune system is going to go dup. It is going to perform very low, and probably at the end later on, the person who have this condition might even die. So what can be done, ma'am? If I think of certain thing, basically there is an enzyme that codes for this particular, there's a gene that codes for this particular enzyme, right? Why not put this gene in a normal cell that does not have it? We can do that. Right, that is recombinant DNA technology. We usually do that. We can take the gene and put it inside our cell. Will that work? Yes, that will work definitely for some time. Suppose I am putting a gene inside the lymphocytes right over here, and now I have taken the gene, put in the lymphocytes, and basically I have it in my body, but these lymphocytes obviously will die after some time. Then what? I will be needing repeated transfusions in this case, but still I will be able to survive if I have this basic thing. So this is known as gene therapy, my dear children. And you are being asked, what are the steps? You have to go step by step. So the first thing that I got to understood from Jasmine is that I have my lymphocytes in my body and I need to put the gene. So for that, I need to take the lymphocytes out of the body. So the first process would be lymphocytes are collected from the blood of the patients. Okay, this would be the first process that I have. And now what? Basically, the lymphocytes now I have collected over here. There might be very few. I cannot collect all of it, otherwise how will I survive? So basically, I take only few, and then what do I do? I culture them in the laboratory conditions. I culture them in the laboratory conditions such that they increase in number. So that will be the second thing that I can probably do. So after the third option, if at all you see only 2 options have third. Rest all of them I can cancel it out. Okay, now third is being followed by fourth over here. The genetically modified lymphocytes are infused back into the patient. No, that is super fast. There are so many steps in between. So let's come back over here. So after third, it is one. What is it? Lymphocytes are grown in a culture outside the human body. Aren't we doing that? Yes, ma'am. Okay, now we have grown them. Now what? We have to put the gene. We have not still put it. For that, what do we need? We need a vector. And please understand, for animals, the usual vector that we have, the thing that will be carrying this gene and putting it over here is usually a retrovirus. Retrovirus, because they know the tactic of how to put the gene inside our cell. HIV, which is a retrovirus, works on the same principle. So, ma'am, are we going to take a virus like that? Don't you think it will cause trouble? We have studied in biotechnology principles and processes that these retroviruses that we're going to take, we're going to disarm them. Disarm them means basically we're going to make sure that they are not going to cause any trouble. We're going to remove all those things which we think can cause trouble. So, this is known as a disarmed retrovirus. Now, we'll put the gene in the retrovirus and the retrovirus will be transferring this gene into what? Our cells. Our lymphocytes. Now, our lymphocytes basically would be having this particular gene. So, lymphocytes are altered with the functional ADA gene. It is altered. Why? And why it is altered? Because we are putting something inside it, right? And what is it? It is the ADA gene that we are putting. Now what? The genetically modified lymphocytes are infused back into the patient. Now, these lymphocytes which are there would be put back into the patient, my dear children. So, in that particular case, what will be your answer? Your answer is going to be C. Your answer is going to be C. Cool. And as I said, the sole way I used over here is to explain your answers, which is why I didn't read the options before, because I wanted you to have a fresh approach to things. Okay, but now the next time when you revise, try to solve this question by yourself. Cool. Now, this particular gene therapy was performed on a 4-year-old child, my dear children, long back, almost in 1990s. And I think I had put the picture. No, I had not put the picture. So anyways, it was being performed long back when to a child who had adenosine deaminase deficiency, my dear children, and it was successful back then. And please understand, there is mostly no permanent cure for treating this particular disease. Okay, the only way you can cure it, there is one, and that is the only way, and that is basically to do this process, that is to put the genes in the embryonic stage itself. When the embryo is being formed, at that particular time only if you put it, then it is totally fine. Apart from that, even if you do bone marrow transplantation, or probably if at all you give enzymes, rather than giving the genes, you directly give the enzymes also. Similarly, you would have to do it again and again and again and again. So the only permanent cure is basically to put the genes in the embryonic stage itself, but that is kind of rare because people generally do not even diagnose it so early. Okay, we do not think about it as such. Okay, so in that case, gene therapy definitely works. It is going to be super helpful. Now, I saw this and it was kind of very interesting to me. This is the bubble boy. So this bubble boy was also tried on his body. Why it is a bubble boy? If at all you have any idea, you can let me know in the comment section, or I'll tell you by myself. So he also had a disease where he could not stand any of the contaminants. So any contamination could cause trouble in his body because his immune system was totally down. So which is why he was always inside a bubble. Inside the bubble, the whole sterilization and everything was being done properly. And then gene therapy was performed on him also long before. Not gene therapy, some other technique was not gene therapy, it was some other technique being performed, and the boy could not survive because the boy's body reacted to it. So his own sister's bone marrow cells were being transported, but still it didn't work because, as I said, it was long back, I think in the year 1940s. So this gene therapy, which was a successful one, it was done in 1990s, and 1940s is pretty long back, right? Which is why it didn't survive. And since I saw that, I thought I should share it with you also. It's like a very, I was so astonished to find why is a child inside a bubble. So yeah, so that is the thing. Now going ahead with the 5-marker question from the model question paper 1. This is question number 40. My dear children, before that, if at all you think that this session is helpful for you, do not forget to hit the like button and subscribe to the channel as well. Genetically modified plants have been developed through core techniques of biotechnology. How are these genetically modified plants useful to humans? Genetically modified plants means, for example, I have some gene and I'm putting it inside. I have some gene that is there for disease tolerance, and I put this gene inside the plant will be disease tolerant then. So that is genetically modified plant, right? Now how is it useful to human beings? That is a question. So you have few points over here which you can refer to and which you need to write. First is enhanced crop yield and efficient mineral usage. So genetically modified plants, they grow faster, they use nutrients better and produce more grains or fruits. This helps farmers get higher productivity even with limited land. Not just live with limited land and also minerals. So if at all you think, if at all there was a plant that needed XYZ for survival, but now it does not need XYZ, but it can survive and survive properly on just X, don't you think it will be a good deal? Because obviously the farmers would not have to buy a lot of stuff around there. So enhanced crop yield and also efficient mineral usage is one thing that help the farmers, human beings. Second is resistance. Resistance to what? Pests and diseases. Now you think, I'm a farmer and I'm investing a lot of money into buying a plant and growing it, providing manure, providing fertilizers. Now if at all this particular plant dies of some pest and things like that, obviously I will be in great loss. Now that can be prevented. How? Genetically modified plants which are there, they are mostly what? Pest resistant, virus resistant, fungi resistant, nematode resistant, and you have examples for it. There is BT cotton. Okay, there is BT cotton. Basically, BT cotton is what? Do you have any idea? So I'll tell the story about that very, very soon. Okay, I'll tell it here itself, and because that is again a 5-marker question that can come up, I have to explain in detail. So please wait for some time, but just understand there is something that is known as BT cotton, where we are putting a gene from a bacteria, which makes the cotton plant disease resistant. And the name of the bacteria from which we are taking the gene is known as Bacillus thuringiensis, which is why it is BT cotton. Okay, now if the plant has this particular gene, any insects which comes and eat the plant will be dying, which is why no more insects will be coming and trying to eat the plant. Now RNAi in plants, that is RNA interference plant, that is another technique. Similarly, my dear children, there is a nematode which is known as Meloidicoin incognita. Okay, so this nematode, a worm, okay, a worm which is there, it usually infects the roots of tobacco plant, which is why the tobacco plant is not able to survive. Now again, with the help of genetic technologies that we have, my dear children, we are able to produce plants which can resist nematodes as well. So resistance to pests and diseases is one such point that we have. Third one is reduced use of chemical pesticides. Because GM plants protect themselves, farmers use less insecticides, my dear children. That means it is going to be less toxic for the soil's fertility also. If at all when you're putting insecticide, it is going to the soil and the soil's fertility also goes down. So in this way, reduced use of of chemical pesticides, and the pesticide cost way too much. So anyway, the plant is pest resistant, so do I need chemical pesticide very much? No. So these 2 points are kind of interlinked. Now, tolerance to abiotic stresses. For example, suppose I have the plant that is tolerant to drought. It can survive with limited amount of water. Kudos, right? Suppose I have a plant that can survive in high saline nature. That means wherever the soil has much amount of salt, still it is able to survive. Usually plants do not survive there. But this plant, which is genetically modified, is able to survive, then it is totally good. So this is also great. This allows the crop to survive difficult climates, and which is why the farmers will be having higher returns. That is how it is useful for human beings. Now, improved nutritional quality. This is one huge thing, and it fascinated me. So my dear children, genes can be added to increase the vitamins, minerals or proteins, and the best example for this is golden rice. Golden rice. Do you know what is golden rice? So in golden rice, it is known as golden rice because it looks a bit yellowish in color. Why? Because over here there are genes in the plant that will be producing vitamin A. That will be producing actually beta carotene, that's a pigment, okay. This beta carotene basically helps in the production of vitamin A. So I can generally say that this golden rice will be rich in vitamin A. Now, why is it useful? So I studied about that, and I came to know that in Africa, there were so many people who faced malnutrition, much more than they had now, okay. They have it now, much more than that it was been there initially. So people thought that if at all, what if we provide them rice, because rice is something that is staple food, and what if we infuse the rice with some vitamin, then probably they will be getting these vitamins as well. You get it? So that is the theory about it. But yes, so there is golden rice which is enriched with vitamin A. Do not forget that. So improved nutritional quality is also one big thing. Now, reduces post-harvest losses. Now, sometimes what happens is that, okay, when it was, the plants were growing, it was totally fine. Now, after we pluck them off, after the harvest, they are not able to survive. The seeds die. The seeds basically are being eaten up by insects and things like that. But now they are being resistant to that also. So genetic modification can slow down what? Spoilage. So these are the points that you need to write for a 5 marker. Write it properly. Give examples for it as shown over here. It is going to help you out really, really well. And as I said, I will be explaining these 2 points later on for BT cotton as well as RNAi interference technology. Okay. Now question number 3. This is from MQP2. And the question number is 12. So, what do you call model question paper 2, question number 12 is the one that is being discussed over here. So, plants developed by the fusion of protoplast of 2 different plants are called as what? So, what is protoplast, my dear children? So, if at all you know plant cells, usually it looks like this. So, they have their own cell membrane. There is their own cell membrane, and apart from that, they also have cell wall, which protects them from everything around it, right? They have an extra shield out there because they cannot move. Now, when I remove the cell wall, when I just have the cell membrane and everything like that, now I will call this content as protoplast. I'll call this as protoplast. And my dear children, suppose I have a cell of potato and I have a cell of tomato. And basically, I'm fusing them. Okay, I'm fusing 2 different plants with the fusion of their protoplast. This will be now known as somatic hybrids. What will be now? They will be known as somatic hybrids, not soma clones, not transgenic plants, not GM plants. Please understand, it will be known as what? Somatic hybrids. Always remember that. So example for this is potato and tomato. So potato and tomato cells were fused. Now you got tomato. So that was an example of protoplasmic fusion, and the things that you get at the end are known as somatic hybrids. Ma'am, what does the other things mean then? So I'll explain that as well. But first of all, let's understand this. When protoplast cells without cell wall from 2 different plant species or varieties are fused together, the resulting plant is known as somatic hybrid. It's a hybrid that is being formed, right? This technique is known as protoplast fusion or somatic hybridization. Why other options are wrong? First one, soma clones. Plants produced from tissue culture, not protoplast fusion. What do you mean by tissue culture? Suppose, my dear children, I have a plant, a normal plant right over there. And if at all you know, the tips of the plant, the shoot apical meristem, the root apical meristem, everything. So there are in the tips, so suppose this is the shoot over here, and in the tip, the cells which are there, it is known as shoot apical meristem. So they have a potency to divide very, very fast, and they can develop into whatever we want. So basically we take those cells out there, we take those cells, we're putting them in some solution which has all the hormones such that these cells, my dear children, first of all they divide, and then later they form the root, they form the shoot and everything, and we get new plants out of it. So that is known as micropropagation. Okay, it is known as micropropagation whenever we are cultivating thousands of plants from these small things. First of all, the process initially is known as tissue culture, where we are culturing them and growing them. But if at all we are producing thousands of them, you will be calling this more elaborately as micropropagation. And now, since all the plants which are being formed will have the same cell type, will have the same genetics, so that is why they are known as clones. They will be known as clones, and specifically soma clones. So plants produced from tissue culture. So tissue culture is a mechanism which we use to produce thousands of plants, which is known as micropropagation. And over here, since all of them are genetically similar, you call them as soma clones. So here somatic hybrid, two different plants are being mentioned. So hybrid has to be there, right? And here it is clone. Now transgenic plants contain foreign genes inserted using genetic engineering. That is known as transgenic plants. Similarly, GM plants are also same, genetically modified plants. Again, not related to protoplast fusion. So the only thing that matches with this option is what? B. I hope you got it. Now briefly explain the uses of transgenic animals. This is a 5-marker question in your MQP2 question number 40. So what is the use of transgenic animals? Transgenic animals means basically they have the genes been inserted from elsewhere. They have the genes being inserted from elsewhere, which is why they are known as what? Transgenic animals. Now, what are the applications for it? First one, to study of the normal physiology and development. For example, I have some a factor, a protein out there, which is known as insulin-like growth factor. I don't know why it moves so fast. Okay, insulin-like growth factor. So now this particular thing is responsible for the growth of our cells. If I directly put it into our body, basically we do not know how it might develop and what problems it might cause. So to understand the nature of it, how does it actually work? What did people think of? People thought of, why not put it inside animals and see how the gene regulation work, how their effects work on development, on bodily functions, everything. So to study normal physiology and development, transgenic animals were super important. It helps in understanding gene regulation and effects on growth, development and body functions. Now, at least some of you might be getting this in your head that this is wrong, right? Well, absolutely yes, it is definitely wrong, which is why there are very strict rules that follows by it. Okay, so it cannot be done randomly on any organism out there. There are so many stricter rules on the basis of which this works. Otherwise, obviously, people will do so many bad things. Now, the point number 2, disease study and human models. So created as models for human diseases like cancer, cystic fibrosis, arthritis, Alzheimer's. So there are some changes being made in them only. Suppose a mice has some changes in their body, we change their genes and things like that, and then probably we treat it with some a drug out there and see whether it is able to treat the disease or not. For example, cancer, for example, cystic fibrosis, for example, arthritis or Alzheimer's. So first understanding or studying the disease is better, or probably not just about drug testing, it is also more about how these diseases affect the body, what all are the problems that are being caused in the body. So that is also being studied with the help of transgenic animals. Now, the third one and the most important one is biological products. So biological products means basically, suppose there is a protein that I need so much, but in my body the production of it is so less. What do? Basically, or probably it is the need of the market. So many people need this particular protein out there. So there are some animals which are made to produce these proteins. For example, there is a disease which is known as emphysema. Please remember it. It is a respiratory disease. Okay, it's a respiratory disorder that is there. Now, for treating it, you need a protein which is known as alpha-1 antitrypsin. Alpha-1 antitrypsin. Now, this alpha-1 antitrypsin is made to be produced by some animals out there. And usually understand the most important transgenic animal is a mice only, because it has so much resemblance in the terms of genetics. Most of our genes are same, only few is different, which is why they are there and we are us. Okay, so yes, it is used for human protein production. Example is alpha-1 antitrypsin for emphysema treatment, which is a respiratory problem. Now the second thing, Rosie the transgenic cow in 1997 produced nutrient-rich human-like milk which has alpha-lactalbumin. So human milk which is there, it has a great protein out there, and this protein's name is alpha-lactalbumin. Now suppose you make a cow produce it. Now the cow which is there, it has the genes to produce this particular protein. Obviously the milk will be enriched with that, and that can be provided to all the other organisms as well. Organisms as in human beings only. So Rosie, the transgenic cow in 1997 produced a nutrient-rich milk which was alpha-lactalbumin. So over here we are making the organism produce what? The biological products as well. Now the fourth one is vaccine safety testing. Transgenic mice. Suppose vaccine safety testing, for example, I do not know if a vaccine is directly inside a human being, how it reacts. So basically a transgenic mice again is used for testing the vaccine safety. For example, polio vaccine was used on animals before the human trials. Now fifth one is chemical and toxicity testing. We do not know how much amount or dose of this particular chemical is going to be safe and which is not going to be safe. So basically what is being done is that in the organism which is going to be tested, they have some changes in their genes such that they show sensitivity, they show more sensitivity, and then this drug is being put down so to analyze, okay, in this much sensitivity, a person has this much sensitivity, then probably the reaction could be this much. So chemical and toxicity testing is also being done. Genetically modified animals used for quick and accurate toxicity testing of drugs and chemicals. So I know all of this sounds cruel. I know all of this sounds cruel, but please understand, my dear children, you are not being taught to be cruel. Rather, these are techniques which are being used. Initially, people used it in a very bad way, but now it has been a lot more stricter, and they are trying to find what is the other way where other organisms are not being harmed as well. So anyway, the question is there about transgenic animals. This is the thing that you have to write. Make sure you give examples such as insulin-like growth factor, what is its effect on growth, and this to study that. So disease study, please write the name of the disease: cancer, cystic fibrosis, arthritis, Alzheimer's. You can be asked what are disease that can be easily studied with the help of transgenic animals. So these are the diseases. Then biological products, as I said, alpha-1 antitrypsin for emphysema, and also alpha-lactalbumin, which is a human milk protein which was made to be produced by Rosie, the transgenic cow, in 1997. Then vaccine safety testing and also chemical and toxicity testing. Done. So these are the uses of transgenic animals. These questions have been repeated throughout the years, so make sure you go through it carefully. Now, also one more thing, as I said, this can be an MCQ question also. Fine. Identify the incorrect statement. The transgenic cow Rosie produced milk enriched with alpha-lactalbumin. I just told you that it can be an MCQ, and there is an MCQ. So model question paper 3, question number 12 is the one that we are dealing as of now. So transgenic cow Rosie produced the milk enriched with alpha-lactalbumin. It is correct. We have to find what is incorrect, right? So whenever you have to find correct or incorrect, make sure you box it up. Sometimes we try to find the correct statements in between all the incorrect ones, so don't do that. Now, Bt toxin genes CRY1AC controls the corn borer. Mama, I do not have any idea what Bt toxin gene. I am there to explain you, do not worry. Now, human protein alpha-1 antitrypsin, okay, is used to treat emphysema. Mama, I know that is correct. We just studied about it, right? Yes. Now, the commercial production of human insulin was made possible by the transgenic E. coli. Do you know that? Well, yes, it was. So I'll tell the story about insulin as well as Bt toxin right here itself, so that you will be able to solve more questions related to it. First of all, let's start with Bt toxin. So for that, I would have to go a bit ahead with the last question of the chapter. Don't worry seeing all the slides, okay? It will be taught like this. Anyway, I hope you're understanding, which is why I am also taking much more time to explain you the things so that you don't forget and you don't lose marks anywhere. So my dear children, here is a question that have come in previous year. One of the applications of biotechnology is to get pest-resistant plants. Justify the statement with reference to Bt cotton. Now you can get a 5-marker question also, which is why I am explaining you in a proper way. So, there is a bacteria that is known as Bacillus thuringiensis. The spelling of it is like this. Bacillus thuringiensis. Okay. Now, this particular bacteria which is there, it produces a protoxin. A protoxin means it is inactive toxin. Now, this is produced by what? This particular bacteria. Now, when I say the bacteria produces it, how does it produce it? Basically, there are genes which codes for the protoxin out there. What do you call these genes as? These genes are known as cry genes, and you will get to know why it is known as cry genes. Basically, it makes the organisms cry. So, it's not exactly why it is known as cry genes. They have been named like that. So, now in cry genes, there are different types of genes which produce different types of protoxins according to different insects it is going to affect. Now, suppose, my dear children, I know that this particular bacteria is producing protoxin. Now, suppose an insect basically has this particular bacteria. The insect actually went to eat a plant, and on this plant basically you had the bacteria right over here. Now, suppose what will happen, it got inside the insect. Now, the bacteria is inside the insect. The bacteria is also able to produce what? Protoxin. Now, this protoxin is inactive version of toxin. It is inactive. Ma'am, why is it inactive? Probably it didn't get conditioned to become active yet. But now, once it is inside the insect, what happens is that inside the gut of the insect, the pH is alkaline. It is basic pH. The pH is alkaline, which is why this protoxin which was there, it will now get what? Active. It will now turn into an active version of the toxin. And now what? Basically, this toxin attaches to the midgut. Okay, the walls of the midgut. So you have hindgut. So you have different types of variations of gut out there. Portions of gut. Now there is midgut epithelium. In the midgut, the epithelial layer is where it will come and perform its action. The toxin affects the midgut epithelial layer of the insect and make pores over here. Make pores in the midgut region, leading to what? Death. Leading to death of the insect. I'll explain once more. So Bacillus thuringiensis, in their body, they are producing protoxins which are inactive in them. Please understand, it is inactive in them, which is why they are able to survive even in the presence of the protoxin. Now, when an insect has it, what actually happens is that the gut of the insect is having alkaline pH, where the protoxin converts into active version of toxin, and now it will cause trouble in the midgut epithelium right over here. It will make pores in the epithelial layer, causing the death of the insect. Now, suppose I know all this thing, I take the gene which codes for this and I inject it in a plant. I put it in a plant. Now, what will happen? If suppose this gene is there in the plant, the same thing applies. When the insect comes and eats the plant, isn't it getting the gene for making the protoxin? And the protoxin will convert into active toxin, and later this particular insect is going to die. Okay, so that is the logic of it. So you have different chi genes. So over here, let's read it. Some strains of Bacillus thuringiensis produce protein crystals, insecticidal proteins. That means the one that kills the insects, that kills the insects such as lepidopterans, coleopterans and dipterans. There are different groups of insects: lepidopterans, coleopterans and dipterans. Now protein crystals form during certain phases of the growth. The cry gene, cry1AC and cry2AB from thuringiensis coding for the toxins are transferred to the cotton plants. Now the proteins encoded by these genes control what? The cotton bollworms, a group of insects which are known as cotton bollworms. I'll explain you all this thing, don't worry. Now BT cotton plants express the BT toxin gene by producing toxin that provides insect resistance. It exists as inactive protoxin, please remember. It exists as inactive protoxin. Insect ingests the inactive toxin, converts to active toxin at alkaline pH of the gut of the insect, which solubilizes the crystals. Now, toxin binds to the surface of the mid-gut epithelial cells, create pores that causes cell swelling and lysis, and eventually the death of the insect. There will be cell swelling because a lot of things will be inserted. There will be lysis, cell will be lysed off, and eventually there will be death of the insect as well. Now, as I told you, what genes for what? So you need to remember that as well. So over here, I told you cry genes are there. There is cry 1AC, 1AC and cry 2AB. And there is one more which is known as cry 1AB. It's simple. Don't be confused. Listen. You have AC, so AC over here, and ABCD, AB is also there. So cry 1AC and cry 2AB are going to produce genes, are going to produce toxins that is going to affect the cotton bollworms. The cotton bollworms. So you can remember C over here. The first letter is C. So the first letter is C and that is cotton. Now the second letter is B, that is bollworm. So cotton bollworms. Now cry 1AB is the one that basically kills cone borers. Now ma'am, here also C and B is there, but this is smaller. That was bigger. So that is why you need two of them. You can remember like that, okay, just for fun. Now over here, how do you remember cone borers? Now suppose ma'am, in the exam I write cry 1AC. Don't write that because over here remember C and B. So cone borers, first letter, last letter, cone borers, okay, and the name is short. Cotton bollworms, think of the name to be long, which is why you need two of them. Here it is short. Once more I'm telling you, the first letter is C, cotton. The second letter is B, bollworms. So you can remember like that for 1AC and 2AB. Sometimes we make it 2AC and 1AB. So that's why I'm telling you again and again. First letter, second letter. Now cry 1AB, corn borers. Just one is enough for that. So you can remember like that. I hope it helps. Now we will come back to what we were discussing here. So where were we? We were discussing somewhere over here. Okay. Yes. So BT toxin genes cry 1AC controls cone borers. 1AC, does it control cone borers? C and C. C should be there, right? So this is wrong. This is wrong. Okay, cry 1AC is for cotton bollworms. Now, so this is the incorrect statement, but we are left with one more statement which you have not discussed. The commercial production of human insulin was made possible by transgenic E. coli. So, do you think human insulin was produced by E. coli? What is insulin used for? Insulin basically is a hormone, okay, which traps all the glucose in our body and converts into what? Into glycogen, such that the level of sugar is maintained in our blood. That is a function of insulin. Now, people generally have, some people you might have seen, the ones who have diabetes, the pancreas do not perform well enough to produce enough amount of insulin, which is why the sugar level goes high. So in them, we need to provide insulin from outside. So how to produce this insulin? That is a big task. So usually initially people used to take the insulin from some pigs or other organisms, but it was some people got allergic to it, which is why there was a need that came that okay, insulin has to be produced from some other resources and it should be human insulin. It should be human insulin. Now how can we do that? Now since we are talking about it, again as I said, So with one MCQ, we are able to discuss multiple questions right over here itself. So just give me a second. So don't think there are so many things and you have to cover all of it. While we solve one question, we will be covering a lot of things here itself. I was talking about human insulins. Where's the picture? Where's the picture? Yes. Over here, I have the picture. So there is a question. Explain the application of biotechnology in the field of medicine. So this is model question paper 3. Okay, not 2. It's model question paper 3, question number 40. Explain the application of biotechnology in the field of medicine. So one is the development of insulin, and the other one is the gene therapy that we just discussed, ADA deficiency, right? So we will talk about that as well. First, over here. So how do you use biotechnology in this particular field is that, my dear children, insulin which you see has A chain, it has B chain, and it has one more chain which is known as C chain. It has C chain. So basically it is made up of multiple polypeptide. One polypeptide, second polypeptide, third polypeptide. Now, if I want an organism to produce insulin, obviously, my dear children, these genes that codes for each polypeptide should also be put in that particular organism. But the problem over here is that usually inside our body, even though the pro version of the hormone, the pro hormone which is insulin, not the mature one, the pro one has A, B and C polypeptide. The mature one which is there, it lacks it. It does not have the C chain. It just has the A and B chain, which has disulfide bonds between them. So, ma'am, the pro hormone which is there, it is not the actual insulin, right? The mature one is the actual insulin. Yes. You are correct. Now, ma'am, in our body, the C chain is being removed automatically by our bodily mechanism, right? Yes, that is also there. So, ma'am, basically then, if we put this particular gene of the pro hormone into another organism, it will be producing ABC gene, and that organism will be cutting off the C chain just like how it is done in our human body? No. And that is the biggest trouble. The organism where we put the gene which is going to code for all this thing are not going to have machinery to cut off the C chain which is there. So what can be done then? So then probably we have to think for other way. That is, we should be taking the gene that codes for A chain, and we should be taking the genes that codes for B chain. And then we should be putting it individually into cells. So basically, one E. coli, suppose it has A chain in them, and the second E. coli, suppose it has B chain in them. I am producing both A and B chain. And now, in the laboratory condition, I have to make sure that both of them come together and they form disulfide bonds. I hope you understood. So in our body's machinery, the C chain is being removed easily. So that is not a problem. But whenever you are inserting in another organism, suppose in E. coli, because E. coli is a transgenic animal that you're going to use. In them, you're going to put the gene for A in one and put the gene for B in another. Now both of them will be produced, and then in laboratory conditions, we have to mix them up. Well, this idea was given by a pharmaceutical company which was known as Eli Lilly, and it is still there. It is a very, very famous pharmaceutical company. So Eli Lilly was the first one to produce the human insulin with the help of E. coli, and this human insulin is known as humulin. It is known as humulin, and always remember, insulin has a pro hormone version and a mature version. It's a pro hormone version and a mature version. Now we'll read out and we will be able to understand it better. So, biotechnology helps produce safe, pure and large quantities of human proteins using microbes. Example, human insulin, which is known as humulin. Earlier, insulin was extracted from cattle, pig, pancreas, caused allergies in human bodies. Now, human insulin consists of A and B polypeptide chains linked by disulfide bonds. Eli Lilly, in the year 1983, created separate DNA sequence for A and B chains and inserted them into E. coli plasmids. So, E. coli has plasmids. E. coli which is there, it has plasmid. Plasmid is something, basically they have their own genetic material. Apart from that, there is some extra chromosomal material we studied in biotechnology principles and processes. Now suppose I am putting my gene of interest over here. This plasmid which is there, it has everything to do self-replication. It keeps on replicating, keeps on producing my protein for sure. So now, bacteria produce both chains which were purified and chemically combined in the laboratory to produce what? The functional insulin. This allows large-scale production of human insulin without immune reactions as well. So please remember, the pro version of insulin is there, which is known as pro-insulin, and then you have the mature form which looks like this. So maturation of pro-insulin into what? Insulin. Cool. Now we'll come back to the question that we were discussing over here. So the commercial production of human insulin was made possible by transgenic E. coli. That is also true. So the only one which is not true is what? B. And while explaining this particular question, you got 5 marks for BT cotton, okay, and also 5 marks for human insulin explanation. I hope you're getting it. Now the next question from the model question paper 3, as I discussed, was this: explain the application of biotechnology in the field of medicine. So we saw in the field of agriculture and things like that, that was BT cotton. There's one more in the field of agriculture, we'll be discussing it later. Now in the field of medicine, the one is production of insulin, the second one is gene therapy for treating genetic disorders. I told you, right? So gene therapy corrects the genetic defects by inserting functional genes into the patient cells. Example, ADNA deficiency, first success in 1990 in a 4-year-old child. ADNA deficiency is caused by deletion. Please remember, this is asked. How is it caused? It is caused by deletion of adenosine deaminase gene, leading to an impaired immune system. Treatment steps: lymphocytes withdrawn from the patient, functional ADA, complementary DNA inserted using a retroviral vector. So as I said, we take the lymphocytes, we grow them in the medium. Please grow them in a medium also. Then basically we are going to insert the gene in the retroviral, first in the retroviral vector, and that retroviral vector will be transferring it over here. So what is cDNA? So that is being mentioned, and that causes trouble in remembering. So if at all you remember, my dear children, what usually happens is that we have our DNA, the DNA undergoes transcription, and then we have our mRNA. Now in our DNA, there are regions that codes for something, that does not code for something. So basically, whenever we form the mRNA, in between that there is another type of RNA being formed, that is known as hnRNA, which has all this non-coding and coding regions. We'll remove the non-coding regions, we'll put just the coding regions, and that is your mature RNA, mRNA or messenger RNA. Now this has only the coding things, we do not need anything else. So what do? From this mRNA, we are going to create a DNA. Ulta transcription, reverse transcription is being done, and we make a DNA which has all the important genes for just coding the protein, nothing else, which is why it is known as cDNA. Complementary DNA. Complementary to what? The mRNA of the original gene. I hope you get it. So the original gene has coding and non-coding regions. We just want the coding segment. So that is that is there in the mRNA. So what if we do reverse transcription? We'll get the entire strand properly. And that is the cDNA, which is there put in the retrovirus vector, and the retrovirus vector transfers it into our lymphocytes as well. Now genetically corrected lymphocytes return to the patient. This improves immunity, but repeated treatments are needed, and lymphocytes are what? Immortal. They do not live for long. So the repeated treatments are needed. A permanent cure is possible by introducing the ADA gene into the embryonic stem cells, or embryonic cells, or the stem cells out there. Apart from that, you cannot find a permanent cure. Please remember, bone marrow transplantation or enzyme replacement therapy. Enzyme replacement means you're just going to give them the enzyme. That also does not work for long. You have to do it repeatedly. Okay? So cool. This question is also explained. This is the 5-marker question. Now, my dear children, we are going to go ahead. We have completed the model question paper questions, and with that we got a lot of information out there. And over here we have the revised question bank questions. This is your PYQs. It contains your previous year model question paper questions, everything. So beginning up with the 7th question out there, this is question bank question number 5 from part A category. So which of the following statement is correct with respect to synthesis of insulin? Now you will be able to solve more questions in a faster rate. So synthesized as proenzyme and needed to be processed to become functional enzyme. So you are asked which of the statement is correct with respect to synthesis of insulin. So synthesized as proenzyme, is it an enzyme? Insulin is a hormone, not an enzyme. So synthesized as prohormone and needs to be processed to become functional hormone. Yes, it is correct. Let's go. Synthesized as proenzyme, again wrong. Proenzyme word is wrong. Synthesized as prohormone and needs to be processed to become functional proenzyme. No. Functional hormone, right? Why do we need this again? So basically the answer for this question would be B. Cool. So I already told you, prohormone, the mature hormone. Now, next question. Crystals of Bt toxin produced by some bacteria do not kill the bacteria themselves because. So Bt toxin, even though Bt bacteria which is there, even though it produces the toxin, does not kill itself with the toxin. Why? Bacteria are resistant to the toxin. Toxin is immature, toxin is inactive, bacteria encloses the toxin in a special sac. What is it? It is inactive in nature, and what do you call it? It is protoxin which is there, or prototoxin which is there. Protoxin or prototoxin which is there. This is inactive version of it, which is why the bacteria itself is not killed. So your answer for this question would be C. It is inactive. It only gets active inside the gut of the insect. Now, question number 9. This is question number 11 of your question bank. Statement 1 says the plants genetically identical to the original plants are called soma clones. The plants genetically identical to the original plants, are they known as soma clones? Yes. Now, soma clones can be obtained through micropropagation. Can they be obtained through micropropagation? I told you, yes. So what do we exactly do over here? Let me see, I have a picture over here. So both the statements 1 or 2 are correct. Now what do you do over here? Please listen to this also. This is known as tissue culture method, and when you're producing so many amount of them, you call this as micropropagation. So you take cells from the shoot apex or from the root apex, wherever the cells are actively dividing. So these cells, the feature of the cells at the shoot and root apex is that they actively divide. So they are known as shoot apical meristem. So I take that and I put it in a primary culture where basically the cells will be dividing more and they will be growing. Then what do? I basically put them in a solution which has hormones, shooting hormones, such that they will be forming shoots out there, and also there will be hormones for roots also. So for rooting also. So then roots will be formed. And now what we do? We take the rooted plant which we have got and we put it for propagation. We put it in a soil, in a proper area, and we propagate it properly. Now imagine you are doing for thousands of plants out there, you call it micropropagation. But anyway, all these plants originated from the first one itself, and the genetic material is going to be same, which is why you call them soma clones. Now my dear children, a question that is being asked usually is that, what are the cells? Okay, why do you take shoot apical meristem cells for tissue culture? So yes, they actively divide, but apart from that, there is also one more function, that is they are usually virus free. Please remember, super important. Whenever you talk about the shoot apical meristematic cells or root apical meristematic cells, they are virus free. So if at all you are asked, if you want to produce virus free plants, from which part of the plant do you take? It is from shoot apical meristem or root apical meristem, Sham and Ram. Okay. Now, next question, question number 10. Pollen grains of a plant with 2n is equals to 48 are cultured by tissue culture to produce a callus. Now, what would be the number of chromosomes in the cells of the callus? So, easy question. Listen, do not go by the terms and do not be confused. Callus is basically when, suppose I took a part of the plant. I took this part of the plant. Okay? It is known as X plant. It was belonging to a plant but first, now it is not, which is why X plant. Okay, so X plant portion is being taken. It is put in a medium where these cells are going to divide. Now these cells are going to divide and going to form something which is known as a callus. So over here you are being asked pollen grains of a plant. You are taking pollen grains of a plant and you are culturing in a tissue culture to produce the callus. What would be the number of chromosomes in the cells of the callus? So whatever were the number of chromosomes in the pollen grain, that will be the same thing which will be there in the callus also. You get it? So over here the plant You need to have some information. The plant which was there, it was diploid with 48 number of 2n means diploid. And normal cells are diploid in nature, right? So they have 48. But what about the gametes? They will be half of it. Half of it. It will be haploid. It will be haploid. So half of 48 is what? It will be 24. It will be 24. So pollen grains, if at all, it has 24 number of chromosomes, then all the cells produced from it will also have 24 number of chromosomes. Remember, pollen grains contains the gametes, which is why they will always have half the number of chromosomes than the normal plant cell. So normal plant cell has 48, it will be having half, that is 24. Okay, so your answer for this question would be B. Going ahead with the next question, statement 1 says Bacillus thuringiensis produce insecticidal protein crystals called Bt toxin. Does it? Yes. These toxins exist as prohormones before entering into insect gut. Protoxin, not prohormone. So again, if at all you do it so fast, you might get it wrong. So this statement is false, this statement is correct. Bacillus thuringiensis produces insecticidal protein crystals called Bt toxin. So that is correct, this is wrong. Statement 1 is true, statement 2 is what? False. Okay, it will be false. Now going ahead with the next question, question number 12. This is a model question paper question of the previous year. DNA in a clone of cells followed by detection using autoradiography is called. DNA in a clone of cells followed by detection using autoradiography is called. So my dear children, usually what happens, suppose I have a segment. Okay. This segment of DNA is known as probe. It is known as probe. Why? Because usually it is being attached to something which makes it visible under autoradiography machine. Okay? Because it has something attached to it. Now, what is the function of this probe is that I know the genetic nature of this particular probe. What is the genetic content over here? Suppose I have so many different types of DNA. I have so many different types of DNA. And now I make the probe go and run through it. It will only go and attach to where there is complementary base pairs. To where there is complementary base pairs. So basically I get to understand that this has the complementary base pair and this also has the complementary base pair. Now you are asked, DNA in a clone of cells followed by detection using autoradiography is called. Basically, the one that is detected by autoradiography is known as what? It will be known as what? A probe. Okay, why is it used? Suppose I did a crime. Suppose I did a crime and my saliva or basically my hair strand, anything was out there, and now I am taking it up. I have taken the, sorry, not me, but the investigating officers have taken the have understood the genetic nature and they have created a probe. Now, the people whom they doubt, they will take the DNA from all of them and basically make this probe run through it. The one where it matches, they will be understood that, okay, that person did the crime. So that is the use of it. So the answer for this question would be probe. So as I said, probe goes and attaches and shows off the color. Now question number 13, this is again model question paper, question of previous model question paper. Use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as. Suppose, my dear children, I hope you know that an American company claimed that basmati rice has to be patent. Okay, you cannot produce basmati rice, you cannot make money out of it. I am going to patent it. But is it possible? No. Basmati rice, everything is produced in India, my dear children. So basically, without talking to the people of India, without giving them the compensatory payment, you cannot put patency on anything. Patency means you cannot, patency means basically if I have a patency on something, that means you cannot do anything with it. Okay, I have the whole rice. That is the meaning of patency. So use of bioresources. I am using bioresources. So not me. Multinational companies are using bioresources, and basically they are not taking proper authorization and also not giving compensatory payment. This will be referred to as what? Biopiracy, biofortification, or bioprospecting, bioprocessing. Your answer for this question is biopiracy. So that is basmati. My dear children, also, how many different varieties of rice is there in our country, India? Please remember that. That can also be a question. It is 2 lakh. It is 2 lakh different varieties of rice is there. It is given in your NCERT. And questions like this also hop up. Somehow it's not there in the model question paper, but it comes. Now, which of the following, okay, the previous year question of March 2023. Which of the following statement is not correct? Most Bt toxins are insect group specific. Aren't they? Yes. The genes which are there, they're insect group specific, right? Now, RNAi, this we have not discussed until now, so let's go with the next one. The recombinant therapeutics do not induce unwanted immunological responses in humans. Does it? The recombinant therapeutics which are being inserted inside our body, it does not induce unwanted immunological response. That is correct. It does not, right? If it does, why will we use it? Now, adult onset diabetes can be controlled by taking insulin at regular time intervals. Can diabetes be controlled by taking insulin at regular time intervals? Yes. If at all we giving the hormone that can trap all the glucose and convert it to glycogen, then why not? So we are left with only one particular statement, and this statement could be the odd one out. RNAi takes place in all eukaryotic as well as prokaryotic organism as a method of cellular defense. Ma'am, I do not know what is RNAi. That is RNA interference. Let's talk about it, and probably we will be finishing up the questions even more faster. So this is the question. This is again a 5-marker question that has been asked previously. I'm explaining you in the form of MCQ also. So what is RNA interference? How does RNA interference help to develop resistance to tobacco plant for the nematode infection? So my dear children, I'll tell you a story. So there is a nematode, a worm, whose name is Meloidigoin incognita. Okay, eat melody, forget everything. Similarly, this Meloidigoin incognita, when it affects a plant, basically it gets inside the cells of the plant and make a big cell around it and take all the nutrition and everything. That is a general function of this particular worm. And usually it affects the roots of the tobacco plant. Now, what can be done to stop this? There should be some way to stop this up. So now, there is RNA interference mechanism. What do you mean by that? So we generally know that in our body, as I said, there is DNA. This DNA knows what proteins has to be made, but it cannot make directly. Rather, it forms an mRNA first, and this is known as transcription. The DNA, whatever information is there in the DNA, it has been put down into a chip, that is mRNA, and this mRNA will be responsible for producing the protein. This mRNA is a single-stranded RNA which undergoes the process of translation to produce the proteins. That is the usual thing. Now suppose you only think, I am interfering this mRNA's work in some way. Basically, this mRNA which is there, suppose I am giving a complementary RNA over here. Do you think this mRNA will be single-stranded anymore? No, it will become double-stranded. Now, if it is double-stranded, do you think it can still make the protein? No. For translation to happen, my dear children, in between the ribosomes, a single-stranded mRNA should be coming in, and the tRNA will be helping in translation and producing the protein. So anyway, if it is like this, proteins are not going to be produced. And this is the exact mechanism that we are going to use. We are going to interfere with the RNA's function. So that is known as RNA interference. And how do we do that? That is the question. So first of all, identifying the nematode gene. Scientists first identify the genes in the nematode that are essential for its survival or ability to infect the plant. Now, suppose I know the gene, that this gene is the one that produces the protein and everything out there. What do? Using agrobacterian tumefacins to insert the gene. So basically, I am going to take that gene up. I'm going to take that gene that is going to form this mRNA and things like that. Okay, I know the gene. I need a vector now to put it inside a plant. Ma'am, why are we putting the gene of this particular thing inside a plant? I'll tell you. What is the reason? So basically, before I go ahead with this, I need to give you an explanation. The gene which I am going to insert is going to produce a double-stranded RNA. It is going to produce an RNA which has 2 strands. This is the gene that I am going to insert. And if I want to insert it inside a plant cell, I obviously need a vector. In the case of animals, it was a retrovirus. It was a retrovirus. In the case of plants, it is usually Agrobacterium tumefaciens. Agrobacterium tumefaciens is the one that we use for this. Now, why this? Because usually Agrobacterium tumefaciens, my dear children, that went off. Okay. Agrobacterium tumefaciens, which is there, it generally has a plasmid. Apart from its own genetic material, it also has a plasmid which is known as TI plasmid, tumor-inducing plasmid. Again, we discussed this in the previous chapter. Now, why tumor-inducing? Because it has a part which is known as tDNA, and this tDNA transfers this particular gene into the plant, causing tumor in them. So, when this Agrobacterium tumefaciens goes in effect, this particular transferred DNA which is there is transferred into the plant cell and causes tumor in them. Now, over here, I will be removing the transferred DNA part. Okay, and I will be putting my gene of interest. Here, my gene of interest is the one that will be producing what? The double-stranded RNA. So, that particular gene is being inserted. The gene that is able to produce my protein. So, basically, not my protein, my content. So, basically, this Agrobacterium tumefaciens, which is now going to act like a vector, has it, and now it goes and infects the plant, and then the plant will be having this particular segment, and this particular segment basically makes it produces what? The double-stranded RNA. The double-stranded RNA is being produced. Now what happens? Now the Melodica incognitia, suppose it comes and does its function, it produces the mRNA which will be later forming the protein. What will be happening? This double-stranded RNA comes over here. They will be splitting up, only one. Okay, out of these two strands, only one, my dear children, would be making up a pair with this and blocking the whole process of protein formation. Does not allow protein formation to occur. So basically, one strand is being degenerated, it is being lost, only one strand is there, and that attaches to the other strand, that is the mRNA of Melodica incognitia, inhibiting from the process of translation to happen properly. So over here, where are we? Here. To deliver the silencing gene into tobacco plants, scientists use Agrobacterium tumefaciens, a bacterium that naturally transfers DNA into the plant cells. So the identified nematode gene is inserted into the genome of the vector, that is Agrobacterium tumefaciens. Now what? This DNA sequence is then inserted into the plant's genome using the Agrobacterium tumefaciens during the infection. As Agrobacterium tumefaciens infects it, it transfers this gene. Now, which transfers the gene to the plant cell. Now, what happens? Production of double-stranded RNA in the plant. Once the DNA sequence is inserted, the plant cells begin producing double-stranded RNA, dsRNA, corresponding to the nematode's target gene. Now, the double-stranded DNA triggers RNA interference. That means it goes and binds one strand of it. Remember, even though it's double-stranded, only one strand of it is going to bind on what? On the on the mRNA and stop it. RNA interference has been done like that, silencing the target gene in the nematode cell. As a result, the nematode can no longer function properly and cannot cause harm to the plant. This is known as RNA interference. It is done with the help of Agrobacterium tumefaciens. It is there for protecting the tobacco plant from Melodica incognitia because it usually causes diseases in the roots of the tobacco plant. Done? Now, this is a 5-marker question. You should know how to write it. Write it like this only. Not make it too complex with a lot of number of information that you have. Be precise out there. And now what is the question that we were solving? We were solving this question. Don't worry, the session is going to end soon. Okay. Now, which of the following statements is not correct? RNA interference takes place in all eukaryotic as well as prokaryotic organisms as a method of cellular defense. Ma'am, we didn't study this. Does it happen in all eukaryotes as well as prokaryotes? Well, it happens only in eukaryotes. Remember, plant is a eukaryote, and in the plant's body it is happening. Okay, so generally in eukaryotes we have a cellular defense mechanism, and that is RNA interference. Our body does that. Okay, now we are using that technology over here for protecting a plant specifically for this particular disease. So it is not for prokaryotes. Prokaryotes means bacteria and things like that. Prokaryotes means the one that does not have a proper nuclear membrane or cell organelles with membranes and things like that. So over here, please remember RNA interference is restricted to eukaryotes, not prokaryotes. It's not a natural, mechanism in the case of prokaryotes. So that is an additional information and a very important information. So each line of your NCERT is super important. So here is the difference between eukaryotes and prokaryotes. As you can see, in our eukaryotic cell, there is nucleus just surrounded by all the membrane and stuff like that. This is the membrane of the nucleus and anything. Here you do not have nucleus, things like that. The genetic material is around there, just like that. Okay? Now question number 14. Match column 1 with column 2 and select the correct options from the following given quotes. Alpha-1 antitrypsin. Remember, alpha-1 antitrypsin is for biological product produced by transgenic animals. It is for treating emphysema. Now, transposon, ma'am, I do not know. Let's go with ELISA. What is ELISA, my dear children? ELISA, do you have any idea? ELISA is actually a detection mechanism for AIDS. We will be studying about that. I have a question for that as well. I have put it that over here. So I've included these questions such that all your concepts are clear from this chapter. Now there is retroviral vector. Retroviruses, where do they act like vector? In gene therapy, because gene therapy is done in human beings, right? Now transposon, as you can come out from their name, they move. Okay, they transfer. So that is why they are known as mobile genetic element. So transposons are mobile genetic elements. Sometimes they can cause mutations as well. So always in match the following questions, try to find A and D first. So A goes with 3. A goes with 3. I have 2 options, like let me cut these options. And D goes with 2. D goes with 2. I just have 1 option, which is why this will be my answer for sure. So B goes with 4. Yes, this is my answer. Okay. Now, as I said, there's a question for ELISA and I can discuss it over here. So ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? And name the disease which can be detected by this method. So what is the principle of this particular technique? Is that, my dear children, suppose, okay, so where am I? Just a second. So over here, suppose a person X has a doubt whether he or she has AIDS. Which is caused by HIV virus. Now, whenever we have some disease, our body will be producing some antibodies against it. Okay, some antibodies against this, and the one that causes the disease, they are known as antigens. So against the antigens, our body produces antibodies. This is the usual logic. Now suppose I already know the antibodies which are being produced in a patient who is having what? AIDS. Now these antibodies are already mounted over here. They are known as primary antibodies. I know it. I have synthesized it on my own because that is how it usually works. These are primary antibodies. Now I take a blood sample. In the blood sample, obviously, if that person is infected, will be having the antigens. Okay, now these antigens are going to bind with these antibodies because they are specific to it. These antigens will only bind to the antibodies which are related to it. So yes, this person has AIDS, but how can I view it? I am not able to view it. For that, again, I'll need another antibody. Now this time, the antibody which is going to come will be attached to some enzyme. And this antibody will specifically come and bind on this antigen. If at all it matches. And as I said, enzyme is there, right? Now what is the use of this enzyme? Basically, I am going to act a chromogenic substance. Chromogenic substance as in the one that will be changing its color in the presence of the enzyme. So basically, chromogenic substance has been added over here, and since the antibodies having the enzymes are locating with the antigens, locking up with the antigens, what will happen? The enzyme would be working on it and will be giving off a color change. Whenever there is a color change, we get to understand that person's blood sample whom we have taken from will be having HIV/AIDS disease. I hope you got it. And the same thing can be done in an opposite way. Same thing can be done in an opposite way. Basically, suppose I have the antigens for HIV. I have the antigens for HIV. Now what do? I take blood sample. From the blood sample, I collect the antibodies. Suppose the antibodies comes and binds over here. How will I get to know? I'll take another help of the another antibody, which is now known as secondary antibody. This is the secondary antibody. Secondary antibody is the one which will come again bind with this antibody. Usually it binds with antigens, but in this scenario, there are different types of ELISA where now this is coming and binding on the antibody itself, and over here it is going to be attached to an enzyme. The same thing happens. You need not worry about this. This is going to cause a huge lot of confusion. Rather, just remember this method itself. So you have the pre-prepared antibodies. You give the blood sample which has the antigens. Now secondary antibodies are there which has the enzymes. You put the chromogenic substance. The chromogenic substance is G, okay, not J. Chromogenic substance gives a color change, and that's how we are able to find it. So ELISA stands for enzyme-linked immunosorbent assay. Enzyme link, because I hope you got to know, it is linked to an enzyme. Enzyme-linked immunosorbent assay. It works on the basis of antigen-antibody interaction. A specific antigen, probably a virus, bacteria, protein, or specific antibody is attached to a plate. It can be either of them, as I told. If the patient sample is added, if the patient has the matching antibody or antigen, they will bind together. An enzyme-linked antibody is then added. A coloring agent or chromogen is also mixed. The enzyme reacts with the chemical to produce a colored product. The color change means the antigen or antibody is present and the disease is detected. If no color change, that means disease is not present at all. So you need not worry about it. Now name the disease which you can find with this is HIV. HIV, AIDS. Not HIV, it's the virus's name. It is AIDS. Okay. Now one thing I wanted to tell you, and that was what? Okay, so you have heard about PCR, like polymerase chain reaction, the one that is used to basically increase the amount of DNA. PCR, polymerase chain reaction, is there for amplification of DNA. Is that method also used for finding any diseases or the pathogens? Well, the answer is yes. So always remember, if at all in your question, a question is being asked, what is that mechanism where it is where even a small amount of pathogen is being detected easily? It is PCR, because in PCR you take that small sample and you amplify it, which is why you are easily able to find it. So that is PCR for detecting small amount of pathogen. Please remember, do not forget it. Okay, so ELISA is different and this is different. It works on antigen-antibody principle, and that is for amplification. Now, why are meristems preferred in tissue culture? I told you meristem cells divide rapidly, but apart from that, they are usually virus-free. So plants produced from meristem culture are healthy and disease-free. That is the most important reason. And with that, I think we have completed everything. RNA interference was already done using agrobacterium tumor fissions, and we also completed the applications of biotechnology, that is BT cotton. Okay, so it's been super done. As I'll just quickly go through if at all I am missing something. If not, then it's okay. Yeah, please go through this also. So this is what I was missing. So what are lepidopterans? Tobacco budworm, armyworm are lepidopterans. Coleopterans are beetles. Dipterans are flies and mosquitoes. So these are the three different categories of insects being discussed. So apart from it, all of it super done. So I hope you will never forget all this information. Now children, if at all you think that you have some doubts which are still persisting, put it down in the comment section. If at all you think that this video was super helpful for you, what do you have to do? You have to subscribe to the channel as well. Wishing you all the best. Please take care of yourselves, and that's all. Bye-bye.